So please start the video.
Good afternoon, everyone. I'm Wei hua Huang from Wuhan University, China. First of all, I would like to appreciate the organizing committee for inviting me to talk about our recent work on stretchable electrochemical sensor for real-time monitoring of reactive oxygen species and reactive nitrogen species signaling and oxidative stress in mechanotransduction. Cells in the body reside in a complex and dynamic microenvironment. Besides the chemical and biochemical factors, as shown in this slide, cells are also surrounded by many mechanical cues. They collectively affect cell shape, proliferation, migration, and apoptosis, and finally the behavior and function of tissues. The process of mechanical force regulating cell is termed as mechanotransduction. It means that cells can sense mechanical forces such as shear stress, strain force, and stiffness, and then convert these physical signals into biochemical responses. The time scale of these events ranges from milliseconds to seconds for the activation of mechanosensors, hours for change in gene expression, days for changes in cell behavior and function, and even weeks for tissue development. To understand the mechanism of mechanotransduction, of course, we can use some molecular biology techniques, DNA sequencing, and mass spectrometry to expand our knowledge about the gene and the protein. However, the cellular responses are triggered upon the stimulation of mechanical forces. In fact, mechanical signal propagation from plasma membrane to nucleus is very fast. Only takes several microseconds. And the conversion of force into biochemical signals occur within 1 second. Mechanical force induced chemical signals are transient and weak at the early stage of cellular responses. So, what about the biochemical response at primary mechanotransduction? In this regard, reactive oxygen and reactive nitrogen species signaling in mechanotransduction attracts our great interest. Because these two kinds of species exist in nearly all kinds of cells and are directly involved in many physiological and pathological processes, which depends on the balance and imbalance between oxidative and anti-oxidative system of cells and tissues. Oxidative stress is a result of overproduction of oxidative free radicals and associated ROS and RNS. However, among the external stimuli that influence the levels of ROS and RNS, the effect of mechanical cues on RNS and ROS signaling are less understood. For electroactive molecules such as ROS and RNS, they can directly undergo oxidation or reduction reaction, and the electron transfer during this process could cause the change of the electric currents. Based on this principle, electrochemical method has fast response and high sensitivity, so the real-time and quantitative detection of biomolecules from cells can be realized. But this powerful technique still faces challenge to monitor the biochemical signals of mechanotransduction responses, because most of the electrochemical sensors are rigid electrodes, and they fail to conformally contact to curved surface and comply with the deformation of living cells. For example, when cells are stretched to simulate the strain force imposed on them, the distance between rigid electrode and the stretching cells is always changing, which will cause possible analytic and kinetic consequences. To solve this problem, our strategy is to develop stretchable electrochemical sensors for real-time monitoring of ROS and RNS and oxidative stress. Here, two sections will be introduced. The first one is the fabrication and functionalization of stretchable electrochemical sensors. And the second one is real-time monitoring of stretch-induced reactive oxygen species and reactive nitrogen species and oxidative stress in mechanotransduction responses. What will be required for stretchable electrodes? Apparently, they should consist of elastic substrate for mechanical deformation and conductive electrode material for electrochemical reaction. Importantly, the electrochemical performance must be stable in dynamic stretching process. In this regard, 1D nanomaterials with large aspect ratios are the best candidate, since they can form continuous network. When the electrode is subjected to strain, one-dimensional nanomaterials could rotate and slide against each other to maintain continuous electronic pathways. According to this principle, we uniformly spin-coated silver nanowires and PDMS film. Then, gold nanotubes were fabricated via mild galvanic displacement between silver nanowires and gold chloride complexes. It can be observed that gold nanotubes and PDMS film form in the form of uniform network, and they have high length, diameter ratio, and hollow tubular structure. When the gold nanotube PDMS electrodes are stretched to a large strain of 100%, no significant change in both electrode resistance and electrochemical performance are observed. This proves that the electrodes still maintain stable electric conductivity and electrochemical performance in a wide range of mechanical deformation. Besides, we have also fabricated a series stretchable electrodes based on such as carbon nanotubes, conductive polymer p-dot, and hybrid nanocomposite of two different nanomaterials. We improve the performance of our stretchable electrodes to meet the requirement of detection of mechanotransduction responses. Considering that in the early stage, there is not enough time for nuclear signaling to change gene and protein within several seconds. Therefore, mechanical force-induced biochemical signals is very transient and weak. Another problem is that cells are easily detaching from the surface of electrode when they are stretched. So we have to enhance their performance in high sensitivity and strong adhesivity. As for sensitivity, we loaded carbon nanotubes on the surface of gold nanotube to increase the electron transport pathway, which greatly improves the electrochemical response to nitric oxide. Also, we evenly coated very small platinum nanoparticles on the surface of gold nanotubes, and this electrocatalyst could increase the catalytic size to the electrooxidation of hydrogen peroxide. To end out the stretchable electrode with strong cell adhesion, phenoboric acid molecules were modified on an electrode to provide a sensor with good site compatibility. Because boric acid group reacts with the thiols in carbohydrates that exist largely on cell membrane. Besides, RGD peptides were employed due to the interaction of its sequence and integrins. Importantly, the modification of these small cell adhesive molecules had a little impact on the sensitivity of the electrode. Equipped with the above high performance, these stretchable electrochemical sensors were used for real-time monitoring of stretch-induced nitric oxide and hydrogen peroxide molecules in mechanotransduction. For cell detection, we could culture cells on the surface of a stretchable sensor, and then a spliced strain to induce a stretch of both electrochemical sensor and the cells thereon. During the dynamic stretching process, stretchable sensor monitors the chemical signals evoked by the mechanical force. Here, I would share some results recorded from endothelial cells and chondrocytes. We all know that the pumping of the heart makes the blood flow circulate through the blood vessels. Blood flow exerts mechanical force on the lining of the vessel in the form of shear stress and circumferential strain. As the innermost layer of vessel wall, endothelial cell can perceive the change in mechanical force and transduce the mechanical signal into biochemical responses, which contain the production of vasoactive molecules such as nitric oxide and ROS. To regulate blood vessel function, also the signaling pathway of two molecules are tightly coupled to maintain endothelial redox homeostasis. However, for a long time, stretch-induced nitric oxide and ROS is difficult to acquire by conventional rigid electrochemical sensors. Now, with our stretchable sensor, it is possible. Firstly, we culture endothelial cell and the stretchable sensor, and then impose strain on the sensor to induce the stretch of both the sensor and the cell culture thereon. The obtained result showed that mechanical stretch could activate endothelial cell nitric oxide synthesis to produce nitric oxide rapidly, and the release amount increased with the strain amplitude. Then, with the aid of a stretchable sensor based on platinum nanoparticle coated gold nanotubes, we found that hydrogen peroxide molecules were also released by endothelial cells immediately after stretch stimulation. Besides, the hypertension and hyperlipidemia models were established, and it demonstrated that the release level of hydrogen peroxide were elevated in this disease than those in normal states, suggesting that patients with cardiovascular disease may be more prone to vascular inflammation. Since nitric oxide and hydrogen peroxide can be well discriminated by applying different potential, that is to say, both hydrogen peroxide and nitric oxide could be oxidized at 0.7 volt, while only hydrogen peroxide could be detected at 0.4 volt. Thus, the nitric oxide amount could also be obtained by deducting from the charge recorded at 0.7 volt. Inspired by this, we further developed dual sensors on the same device for simultaneous monitoring of stretch-induced transient hydrogen peroxide and the nitric oxide release from undersized cells. Quantitative analysis revealed an increase in ROS production and a parallel decrease in nitric oxide bioavailability and pathologic conditions, which lead to endocyte redox imbalance and homeostasis alteration. Another typical tissue that bears mechanical force is articular cartilage, and joint function or disorder is closely associated with mechanical loadings. Generally, NF-kappa B signaling is activated in chronic inflammation to produce downstream nitric oxide molecule. However, whether mechanical stress induced nitric oxide production in very short periods was unclear. By culturing condensate on a stretchable sensor, we compared the cell responses when they are stimulated by different strains and inflammatory factor interleukin-1 beta. We can clearly observe that nitric oxide release was evoked upon the stretch loading, while nitric oxide release stimulated by interleukin-1 beta was recorded after several minutes. Therefore, our result indicates that mechanical stretch could rapidly activate NO signaling system to produce nitric oxide. In summary, stretchable electrochemical electrodes have been fabricated based on one-dimensional nanomaterials and functionalized to enhance sensing performance. And reactive oxygen species and reactive nitrogen species and oxidative stress in cell mechanotransduction were monitored in real time by these stretch sensors. In addition, we also summarize the very recent progress in this area in 2 review papers. Finally, I sincerely appreciate the contribution of our collaborators and the students, and also the financial support from NSFC and Wuhan University. Thank you very much for your attention.